Uh, hi, hi everyone. We will be just continuing the little discussion what we had on the previous day on mutation. Let me just take up a little work today. When I was going through the web on the content about mutation, I came across a flow chart. That flow chart I thought is very good as far as I am concerned because I've been reading biology books after 1987. So I thought I should be able to share with you. So I have just taken that and expanded a few things here and there. So we will try to learn and continue the discussion on mutation. So today we will just examine what's called the mutation. What do we mean by mutation? This word normally in genetics, cytogenetics or pharmacogenetics, cellular biology, biochemistry, recombinant DNA technique, anywhere you go, you get the word sometimes mutation. Then once we get that word mutation, normally it is referred to the genetic material, and that genetic material which will be responsible for a change, and that change may be inherited to the next generation. So the word mutation simply means changing. Mutation refers to a process of changing. What is that going to change is a question. In this case, the change will be in the genetic material called the DNA, and if you follow central dogma, DNA change will reflect in mRNA. mRNA in turn will pass the change to the proteins, and proteins in turn will pass it on to the organism. That's how the whole link goes into mutation. So in simple terms, mutation we mean a change. Change is possible in the genetic material either naturally or by means of any mistake or error that happens during the cell division, cell cycle, especially in cell cycle. That is the most important thing where the change can happen. So either an error while transcribing the DNA material, or while translation, or maybe at the base level, because the entire cell, if you look at the cell, is a very minute thing, and inside the cell we will be talking in terms of angstrom units. That means that will be millionth of a 1 billionth of a inch. That is the size we are going to talk about. Even the mutation, you can understand what is the amount of work scientists have been doing on these things. Well, let us forget about that one. Let us come back to mutation now. So mutation can occur due to error or a mistake in the genetic material. In the genetic material, we take DNA as one and RNA also another one, because some of the viruses and also some of the prokaryotes may have RNA also with them as the genetic material. That also brings in changes. Mutations are normally non-fatal, but there are some mutations which are fatal. Mutation can change the phenotype. Mutation can change the functions of the organism itself, or the part, or the protein, and it also changes the structure of an organism where the genetic material is involved. That is the broad way we look into the thing, and the product will be different. So when the mutation happens, that means a change in the DNA or RNA, and that will produce a different material. It may be a protein, it may be an organ, it can be an organ system, it may be a function, and that also will show changes. And that is possible due to gene expression, that you may be able to learn at a later time. So today we will take up a flow chart. And that flowchart is very interesting because I told you I was impressed by that flowchart, so I am presenting it to you. You might have already seen that. but still I would like to discuss with you. In this flow chart, we are going to talk about what's called the point mutations. Mutations happening especially on one nucleotide. You know, the nucleotides will contain the base pairs. The base pair will have or will undergo a change, and that change may be either induced or spontaneous or natural, or it might happen due to accident in the cell, or error in replication, addition of new thing, deletion of new thing, substitution of something. Any of these things might happen to bring a change in the genetic material. So now that's the point we are going to call point mutation. And now in the point mutation, when you say it happens especially at the codon level, if there are triplet, for example, you may have example A, T, T. If this is the triplet code of a codon on a gene, on a segment of a DNA or a part of a DNA, where what is called the coding part of the DNA. You know, there are two parts of the DNA, sense and also the sensing one, and then the no sense one. That means the one that is going to, one will be a template, other will be a real one. So now this ATT, this base pair, one of them, you know, there are base pairs, means it may be adenine, thymine, it may be cytosine, guanine. Any one of them, if it changes or it is deleted or it is substituted or addition of a new thing might bring in the change in the product that is in the protein itself. Let us try to know. See, in this case, please try to look at, there is a lot of diagram, a lot of words are there. Please don't look at all those things. We will take you bit by bit. Now when we come to that, we have a coding one that's called sense strand. That is the one which will have a complementary strand called antisense strand. There are two strands in the DNA, and that forms helix-like structure later on. And then whatever the sense strand has, it will have a complementary base on the other end, at the 3'5' end, that's called the antisense end. On this antisense end, or what's called the template of DNA, there will be the mRNA will be produced. This will be copied, and that's called, you call the transcription. So transcription goes on one strand of the DNA. So it is here, either a change might happen either in the sense strand or in the antisense strand or in the mRNA. Anywhere of these three, if a change happens in the base or in one part of the base, then definitely that there will be a change in the protein, structure, function may change. We shall look into that with one example. Here is one very simple one. I have told you. Here we have taken 3-letter codon. If you look at it, 1, 2, 3, 4, 5 codons I have taken. In this case, a codon normal DNA template. On this DNA template called TTC, thymine, cytosine. On this one, they usually that will be the transcript will be adenine, thymine, and guanine. You know, wherever thymine is there, you will get the adenine. You will get it adenine, etc. So this will be on the codon on the mRNA. So this is the template. On the template, it has been now copied. We will take only, we will forget this. We will take only this particular part, and this particular the mRNA sequence AAG will will produce lysine as one of the amino acid, picks up one amino acid. So if you take TTC in normal form, 1, 2, 3, 4, 5 of them, we get 1, 2, 3, 4, 5 lysine, we should get it in general. But for the sake of understanding, I have shown in only one of them, but the chain I have shown. If there is no change in the TTC codon, if all of them contain TTC codon, you will get a sequence of 5 lysine chain, polypeptide chain. That is a normal case. Okay. Now we come to the changes. A point mutation might happen at point number 1. Here I have shown you here this one. This is the one I have shown. On 1, instead of cytosine, let us say thymine is replaced or substituted. It is changed. So that means there will be a change in one of the sequence of a triplet code. In this case, we have taken thymine in the 3rd position here. That change has taken place. If that change is there, then what happens is, then correspondingly you will have on the mRNA A as the codon in the mRNA. Now this will, the tRNA will contain U, and it picks up again a lysine, because you know what's called degenerate nature of the DNA is there, in that the one that is many amino acids can be picked up by one particular codon. That is the way it is. If valine, there are, see, if the valine is there, valine can be picked up by 5 or 4 combinations of the codon combinations. It's possible. So that is called the degenerate condition of the DNA. So here in this case, this particular AAA will also code and pick up the lysine. So in this condition, in the case number 1, where cytosine is replaced to thymine, there is, though there is a change in the base here, it is copied now to anticodon, and the tRNA picked up the lysine, and there is a lysine has come to this position. There is no change. That means this, though there is a change in the sequence of the base of the mRNA base at the mRNA level, but there is no change because the same amino acid is produced. That is called silent point mutation. There is no change in that. Now let us take up the second case. In the second case, I have shown here adenine is replaced in this position. Thymine that is present in 3rd position here is replaced by adenine here. The first one, the first case. Here we have taken the 3rd base, we have taken here the 3rd base. When that is changed to adenine, now adenine in its anticodon will have UAG, and this will pick up, this is the codon, this will pick up the that is ATC, that will pick up UAG. Now this UAG is a stop codon, and therefore no amino acid can be picked up here, and the lysine will not be there. So there is a break in the chain here. So we just replaced one of the bases, and that base is the thymine is replaced to adenine in the mRNA, and then we got here the UAG as a complementary base for this particular base pair here, and this UAG when this tRNA picks up, there is no anticodon for it. therefore it will be stop, that breaks. And that kind of point mutation is called nonsense, because the polypeptide chain will not have any meaning unto that one. That is called the nonsense point mutation. In the next case we have taken, now I have taken the second letter or the second position where a base is there called the thymine, and this is on the DNA template. Remember, this is the template one, it's not the original, it is not the sense codon, it is the antisense codon has given this particular sequence here, and for that there will be the sequence in the mRNA will be UAG. UAG has come up, sorry, here we are taking about the second one, thymine. This is the thymine here. Thymine is replaced, I have told you already, it's over now. In the second position, thymine is there. In this case, the thymine is replaced by cytosine. Thymine is replaced by cytosine here. When cytosine is replaced, that will form the base anticodon in the mRNA will be AGG. AGG. That will be picked up by the tRNA, and for that you get arginine as a sequence. Now instead of lysine, here we get the arginine. So this is called missense. This arginine has no meaning in the total polypeptide chain, because it has only come as a part of it. It has no meaning for it. That's called the missense. Take another example of missense, the same thing, that is in the second position, the thymine base is replaced by guanine base. When that happens, you will have the anticodon ACG, then you will get here the threonine will become, and that will pick up lysine. So in the missense there may be two types of changes. One is called conservative type. Conservative type means where an amino acid is replaced which might resemble the original amino acid in many respects. So the changes in the protein may be very small, may be unnoticed. That's called conservative. It tries to conserve though the base change has taken place. But in the second case, the base is changed from thymine to guanine, and that is changed, the entire the amino acid is a different one. We get threonine. This threonine has no similarity to lysine. You get the threonine. So this is called non-conservative type, where the original character is not conserved. So this is a simple example where we can study that a point mutation can bring in many changes. One more example I have taken here, just for the sake of understanding. In the normal hemoglobin, when the normal blood cells are taken, that will be discoid or circular, because it will contain 4 possible protein molecules, and that will form a complex, hemoglobin complex, and in one of the polypeptide chain of the hemoglobin, there will be what's called CUC, CAC. CAC normally produces glutamic acid. CAC goes to GUC, GUC will bring glutamic acid. Supposing the cytosine is replaced by guanine, then what happens is this will become GUC, and that will bring the valine. That is the one that will change the protein structure, protein function, everything changes, and then people may suffer by a disease called sickle cell not the disease, a disorder of the blood called sickle cell anemia. Anemia means bloodlessness, because when the hemoglobin, when the blood cell becomes sickle cell like that, it loses a large amount of the portion, and therefore oxygen cannot be carried. So that is one example also for the missense mutation, because here instead of cytosine it has been replaced by guanine. Same thing might happen in a way by addition or deletion of the particles. Supposing we will take up now here we will rub it off. I'll just rub it off, and then we just quickly take up another one. Supposing we add here, for example, it is TTC, 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 TTC. If this is the codon we have on the DNA template. So in case if we add one more here, a new thing is added here, for example. Now this will be all right, TTC. Now this becomes ATT, this becomes CTT, this becomes CTT, and this like that. So now here the entire frame of the structure of the protein has changed completely, and then this is called frame shift. Though only one of the bases added, here it is addition, then that will be seen in the production of the protein. Supposing we take the same thing, and let me say instead of this one, what I do is I will remove this C. When you remove this C, then what we get is we will get TTT, then you get TCT, then you get TCT, like that. So here this is the deletion. So if one base is deleted, then also the frame shift takes place, and this frame shift will give a different type of a amino acid, and that may not be synonymous with the original one. Then the entire structure will change. So like this, point mutation may be fatal, as it in the case of the sickle cell anemia. Sometimes it may be beneficial. When it is beneficial, supposing we take the cancer producing cells. If you take this cancer producing cells, you know a lot of cells are produced. If you can add the stop codon genetically by means of intervening with the protein synthesis, then the stop codon will stop the production of the proteins, and then cell is not formed. That may be beneficial. So like that, in a cell machinery, always there will be changes going on. Since the DNA is very stable, changes will not happen normally, but on some occasions changes might happen. You must have seen the movies in the Nagasaki and Hiroshima bombarding, and then when the generations came out later on, there were people without nostrils, there were people without the earlids, like that, and such mutations are all due to mutagens. Mutagen may be a chemical, mutagen may be a radiation particle, anything could happen. Then also mutations will happen. To end up, we say mutation is a change, and that change happens in the genetic material. It may be at one point, especially involving only one base, or it may involve the entire segment of a DNA, that is a large scale one, or it might involve one chromosome, or it may involve a segment of chromosome, or it may involve many chromosomes. So anything is possible, and whether it is one segment, two segment, or the entire chromosome, there will be a change in the next generation, and therefore we say that has been mutated organism. So mutation will cause mutations. It may be beneficial sometimes, it may be fatal sometimes. We shall stop here and re-examine this chapter in the next session, please.